Hi students, so today we're going to be learning about the respiratory system, and like I've done with many of the systems we've learned about already, I'll go over the functions first and then we'll look at the specific structures and then their specific functions. Last time we left off on the digestive system, which we began learning about body tubes and the layers of body tubes. And I mentioned in that lecture that you will see this pattern of layers of tissue in all body tubes from here on out, in all the systems that we learn about from here on out. All body tubes have these multi-layered organs that have a lumen or a passageway on the inside that are lined with specific tissues, and then when you go from deep to superficial, those tissues change based on the function of those tissues and therefore the organ as well. So we'll get into that here in a minute, but I just wanted to start out with that reminder. So functions of the respiratory system are going to be those that you already know. So for instance, providing an area for gas exchange, that's the ultimate goal. We want to exchange CO2 and oxygen. Actually, that's not a two-way arrow. We want to get rid of CO2 and pick up oxygen. And that's going to be occurring inside the terminal structures within the lung. And we'll get to what those are in a little bit. But those terminal structures in the lung are really well vascularized. So CO2, as you might recall, is a byproduct of cellular respiration, right? So inside of all of your cells, you're taking some glucose in the presence of oxygen, and the ultimate goal is to make ATP, but byproducts are things like water, CO2, and of course, as you know, heat. That CO2 diffuses into our blood vessels, and we can't allow that CO2 to build up in our blood. We've got to get rid of that. So every time you exhale CO2. And every time you inhale oxygen. So this is what we mean by gas exchange. Another function of the respiratory system, in order for that gas exchange to occur, we've got to move air into and out of the lungs. And so muscles are going to be responsible for that primarily. And along the tubes that make up the respiratory system, those tubes are going to be made up of a mucous membrane. So they're going to help to moisten that air as it moves into the lungs. Ultimately, the way gases, the CO2 and oxygen are exchanged is through diffusion. And diffusion can't occur over a dry surface. So moistening or humidifying that air is really important. The respiratory system also warms air as it's being taken down deep into the lungs. If you think about it, we like to stay at somewhere around 98.6 degrees. And at 98.6 degrees is metabolically expensive to maintain, meaning it takes a lot of energy for us to hover right around 98.6 degrees. And in our ambient environment, unless you're in a very tropical location or out in the desert, rarely is it 98 degrees in the air is rarely 98 degrees surrounding you. So if you think of the temperature in your house right now, maybe it's 70, 71 or something like that, that's like 27 degrees cooler. So if you think about it, you're breathing in air that's somewhere maybe around 20 degrees cooler than what your body temperature is. So as that air makes its way into your nasal cavity, into the nasopharynx, down the pharynx, into the trachea, it's gradually being warmed up, so we don't have to spend a whole lot of energy or excess energy keeping our body at 98.6. We don't want the air that we inhale to cool our body so much that we have to spend excess energy warming it back up. The entire respiratory tract, as I mentioned earlier, is made up of a mucous membrane, and that mucus, as you know, traps pathogens so that the mucus then kind of makes its way out of the body, and that mucus, of course, then will have those pathogens, debris, microorganisms, whatever, trapped in it. So that mucus is meant to come up and out of the body. Part of the respiratory system is your larynx, or what you might refer to as the voice box, that helps us to create different sounds. The other function, or another function of the respiratory system, is the regulation of pH of body fluids. And let me see if I can find a place to write this here. Earlier, I mentioned that CO2 is a byproduct of cellular respiration, right? But anytime you add CO2, carbon dioxide, to a watery solution, it turns that solution a little bit more acidic, or it lowers the pH of that solution. So let's say your cells are getting rid of CO2, and that CO2 is ending up in your blood, which is a watery solution. When CO2 builds up in your blood, it decreases the pH of your blood, making something, or an intermediate molecule called carbonic acid. And it goes back and forth, but via carbonic acid, and it will lose a hydrogen and gain a hydrogen. That's something you'll learn about in physiology. But the point is that as your cells release CO2 and that CO2 gets into your blood, pH drops. And that's a problem. We like our blood pH to be somewhere around, hovering around 8.2, or a little bit less than that. So we can't allow CO2 to build up in the blood, otherwise it will disrupt homeostasis of other functions. And then lastly, this semester we didn't get to talk about it, it's kind of a bummer, but inside the nose you've got special sensory receptors that allow you, or give you a sense of smell. But that's ultimately another function of the respiratory system. So just real quickly, if we go over an overview of the respiratory system, this is the route we're going to be taking. We'll be looking at the nasal cavity first, and how air travels through the nasal cavity, down through the nasopharynx, then through the oropharynx, down into the larynx, down into the trachea, down into the primary bronchi, then secondary, and then tertiary, and then all the way out to the bronchioles, which are these branching structures. And at the very tips of those bronchioles are going to be these little grape-like sacs called alveoli, and that's where gas exchange is going to occur. So that's kind of the route of that we're going to take. So again, remind yourself that this is a system of body tubes that are carrying air down to the gas exchange surface. If we erase all of that here real quick, I want to just bring something to your attention. Clinically, I guess not even clinically, just in your daily lives, you can see that the respiratory system is broken up into what we call the upper respiratory system and lower respiratory system. So that dividing line is right here. So here's the esophagus, immediately posterior to the trachea. Here's the trachea anteriorly. So right where those two structures begin, that's the dividing line between the upper respiratory system and the lower respiratory system. So all of the structures that are above the esophagus and the trachea are referred to as upper respiratory system structures. And then everything below, including the trachea and all the bronchi, and of course where we find those alveoli, those are considered to be part of the lower respiratory system. So if you're not feeling well, you go to your doctor says, oh, hey, you have an upper respiratory tract infection. That means you've got some sort of pathogen that has invaded the structures of the upper respiratory system, maybe even your sinuses, like the frontal sinus, and is causing you those symptoms. Lower respiratory infections are things like bronchitis, pneumonia, COVID-19, which is caused by the coronavirus, as you probably know, would be considered a lower respiratory system illness. It attacks the structures that are actually responsible for gas exchange. So that's just a real quick overview of how we can divide the respiratory system up into upper and lower respiratory systems. So if we think about one of the main functions of the respiratory system, that's gas exchange, right? And gas exchange is the exchange of CO2 for oxygen. But in order for gas exchange to occur, we've got to get air down to where that exchange occurs. So there are a number of structures along the way in the respiratory system that are just meant to get that air where it needs to go. And all of those structures are what are part of what we call the conducting zone. These tubes are conducting air where it needs to go. So the conducting zone goes from the nasal cavity all the way to the bronchioles. And again, we lost some formatting there. I apologize. But that's going to be the focus of this first part of this lecture, are structures that make up the conducting zone, meaning these structures, these parts of the tube, do not carry out gas exchange. They're assisting in bringing air down to where gas exchange would occur, or moving air up out of the respiratory tract and out through your mouth and into the ambient environment. Like all body tubes that you've learned about so far, and you've heard me mention this over and over, if you leave anatomy with one piece of information, know that all body tubes are lined with some sort of epithelial tissue. The type of epithelial tissue that lines a body tube will vary based on the function of that body tube. So if we look at the structures along the conducting zone, you'll see that the epithelial tissue varies because these structures' functions will vary. So I'll go over all these structures with you, but let's look at the vestibule, for instance. The vestibule is lined with pseudostratified ciliated columnar epithelial tissue. The vestibule is this wiggly, fleshy bit of your nose right there, lined with pseudostratified. If we continue on, moving through the tube, this section right here is what we refer to as the nasal cavity proper, and that's also lined with pseudostratified. And then this region right here, which we call the nasopharynx, that's also lined in pseudostratified. Leading away from the nasopharynx, or inferior to the nasopharynx, is this next section of tube, and we call that the oropharynx. And here's where you really see a change in the epithelial tissue. It's now stratified squamous. Well, think about what's passing through there, right? Food and drink are going to be passing through the mouth. They could be carrying toxins, or they could be particularly abrasive. So when food or drink makes its way into the oropharynx, it's best to have the oropharynx lined with stratified squamous for protection. From there, then, the tube gives way to, or becomes, what we call the laryngopharynx. That's also lined in stratified squamous. That food or drink will be continuing on. And then if it's air, if you're inhaling air, it should go anteriorly down into the trachea. Food will continue down on and go down the esophagus posteriorly. But this is a shared pathway right here for air, food, and drink. So that laryngopharynx, for protection purposes, is still lined with pseudo-stratified. Let me start erasing some of these colors here for you. The trachea, which will be the next most distal or most inferior section of the respiratory system, is going to be lined in pseudostratified also. And you can't see it on here, but as we continue distally, the not only the trachea, but the bronchi are also lined in pseudostratified ciliated columnar epithelial tissue. And then you finally get down to the very last structures of the conducting zone, not the final structures of the respiratory system, but of the conducting zone, and those are called bronchioles. And here's where we really begin to see a change in the epithelial tissue. First, we'll see in the beginning of the bronchioles, in the proximal most portion of the bronchioles, we find simple columnar epithelial tissue. And then in the most distal portion, we find that simple columnar changes to simple cuboidal. So imagine here in the bronchioles, we're about to approach the respiratory zone, or where gas exchange occurs. So it makes sense then that the epithelial tissue begins to change its structure in order to accommodate gas exchange. We're going from a tall columnar cell down to a shorter cuboidal shaped cell. And ultimately, where gas exchange occurs, you'll find that it's an even thinner surface made of simple squamous epithelial tissue. So we'll begin with where air enters the respiratory system. Actually, there are two places. Obviously, you can breathe through the mouth, but we'll start with the nasal cavity. We'll start with the different regions of the nasal cavity. Air will first enter through the external nares, and that's what we would refer to as the nostrils. And then this region right here is what we refer to as the nasal vestibule. And so that's kind of like the wiggly part of your nose. The next region is what we refer to as nasal cavity proper. And recall, this is where we find the nasal conchae. And those nasal conchae are bony structures, but they're going to be covered in a mucous epithelium made of pseudostratified ciliated columnar epithelial tissue. These nasal conchae function to slow down air as it makes its way into the nasal cavity proper, slow it down, warm it up, and humidify that air. And then as we continue posteriorly, that nasal cavity proper then will give rise to this first section of the pharynx that we call the nasopharynx. And the entrance to the nasopharynx is what we refer to as the internal nares, or your internal nostrils. So here's that nasopharynx on this slide right here. And that goes from the internal nares all the way down to, if you recall, the structure called the uvula. That's the tip of the soft palate. And now that nasopharynx is going to communicate or connect with the oral cavity. And so this next section of the pharynx is what we call, and it goes actually over to the hyoid bone, but it's actually just this section of tube here. But I'll draw the line over the hyoid bone so you recognize where that is. That's the what we call the oropharynx. And this is the section of the tube that's immediately posterior to the mouth, and it's lined with stratified squamous epithelial tissue. That stratified squamous epithelial tissue is going to protect the underlying body tissues from anything that you may ingest through the mouth. So food or drink coming in through the mouth might be abrasive, it might have pathogens in it or debris, and so the stratified squamous that lines the oropharynx serves a protective function. The next and last section, and a much smaller section of the pharynx, is what we call the laryngopharynx. That's also lined in stratified squamous because this is still a shared tube. Remember, anything that you ingest is going to pass through that oropharynx, pass through the laryngopharynx, and if it's food or drink, it should go down the esophagus. However, if you're inhaling through the nose or the mouth and it's air, that air should go down into the next section of the respiratory tract, which is what we call the larynx. The larynx is also known as the voice box, and it's this structure that you see here. And it's really made up of cartilages that you see here in blue, and then what you see here in that beige color is essentially connective tissue. However, this right here, that's the hyoid bone. So that's more specifically bone. And this larynx then is going to be essentially made of different pieces of cartilage that support the structure itself. But not only that, those cartilages are going to, and some of the connective tissue there, are going to be responsible for producing passing sound. So if we have a closer look at the larynx in this picture from your textbook, you see over on the left there's an anterior view, and over on the right that's the posterior view. Now the larynx, being part of the respiratory system, yes, it is a structure that allows for air to pass through, but don't forget that there are structures in there also that are responsible for creating sound as air passes over or near them. So we'll get to that in a second. So let's look at the cartilages that make up the larynx. There are 9 pieces of cartilage that come together to make the larynx, and then ligaments and muscles that hold those cartilages together. So if we first look at the thyroid cartilage, there's only one of those. This is not paired. That's what the number in front means. So the thyroid cartilage is anteriorly located right here. And then the next cartilage is what we refer to as the epiglottis. And from an anterior view, it's a little bit difficult to see. It's this little flap of cartilage, but it's better or easier to see from a posterior view here. And what the epiglottis will do is when you swallow a piece of food or have something to drink, that food or drink hits the top of the epiglottis and it folds down over the glottis so that food or drink doesn't make its way into the trachea. So it kind of diverts that food or drink down into the esophagus. The next cartilage is what we call cricoid cartilage, and again, there's just one piece. So from an anterior view, it almost looks like a Batman signal, doesn't it? Or symbol, not signal, symbol. And posteriorly, you can see it here, so it kind of wraps all the way around the trachea. And then the paired cartilages, where there are 2, one on either side, there are the arytenoid cartilages, and that's what you see here in yellow from a posterior view. And then there's what we call the corniculate cartilage, and corniculate cartilage are the little horns on top of the arytenoid cartilages. And then lastly, they're what we call the cuneiform cartilages, again only seen from a posterior view. Oops. And you can see that here, these pieces. So let's go ahead and have a look at the functions of some of these cartilages. The thyroid cartilage that you see outlined in blue protects the airway from the anterior side and from the walls or the lateral sides of the airway. The epiglottis, as I mentioned earlier, folds down over the glottis or entrance to the trachea when you're swallowing food or drink. And then the cricoid cartilages posteriorly protect the posterior wall of the trachea, or posterior wall of the airway, and it also anchors the larynx to the trachea. Okay, students, so in this picture you're looking from a superior view into the larynx, and the authors are showing you two things here. This is where the, this is where in picture A right here, you're looking down into the larynx with the glottis open. And the glottis is the passageway into the larynx. So it's not a physical structure necessarily. It's not a something you can grab. It's a passageway. It's the entrance to the trachea. So you're looking down through the larynx into the trachea. In fact, you can kind of see some of the tracheal folds right here immediately behind it. This is anterior, posterior is here, and here is that epiglottis. And as you look down into the larynx, you see two folds. Vestibular folds, those are also called false folds, and what we call they're saying vocal fold. You would call those the true vocal folds or the true vocal cords. So let me use the highlighter here so it's easier for you to see. The vestibular folds are these membranous structures right here. And the vocal cords, or the actual connective tissue structures that vibrate when we make sound, are here in green. So the vestibular folds aren't actually contributing to vocalization. It's the true vocal cords. So that's why we call them the either the true vocal cords or vocal folds and the false vocal folds. Because the trues are the ones that are making, they're the ones that are vibrating as air passes by them to create sound. I think there's another picture that I'll get to showing you that here in a sec. Let's go ahead and then look at picture letter B right here at the bottom. And what that's showing you is really the same view, but with the glottis closed. So again, here are the true vocal folds. Those are the ones that are going to vibrate and make sound, remember. And then off to the side here and here are those vestibular folds. This is a little bit difficult to understand that out of order. So we just finished up learning about the larynx and the cartilages that make up the larynx and the vocal folds that cause or allow us to make vocalizations. And now what we're moving into is this next section of the conducting zone, which is the trachea. I'm not sure what happened to all of my text over here, and it's okay. We don't need that for now. So now we're looking at a tubular structure that has a couple of features on it that you can see from this view. First of all are the tracheal the tracheal cartilages, not the folds. And those are indicated here in blue. That's hyaline cartilage. If you remember way back when we were learning about tissues, hyaline cartilage, one of the places hyaline cartilage is found is surrounding the trachea. So that's what those are. What those tracheal folds are doing, or those tracheal cartilages are doing, is they're helping support the trachea upright, so they're giving the trachea structure, but they're also holding the trachea open full time. I'm not sure if I mentioned it in the digestive system lecture, but the esophagus collapses on itself and is only open while food and drink are passing through it, and that prevents pathogens from having a direct open door into the body. Well, the trachea is a little bit different in that if you're not inhaling, you're exhaling. So the trachea really needs to be open full time in order for gas exchange to occur. So those tracheal cartilages are holding that trachea upright in the thoracic cavity, but also holding that trachea open. In between those folds, the beige area, that's just connective tissue. You don't need to know what kind, but it's the connective tissue that's creating the remainder of the trachea itself. Okay, So this is out of order. So I was just saying that those pictures that we looked at from our textbook were kind of a difficult view. So I screenshotted some of the pictures from the Complete Anatomy app. And let me just show you. Here you're looking at an anterior view of most of the respiratory system from the larynx on down. And just to give you a landmark here, that would be the thyroid cartilage. And this is the trachea right here. So if we were to rotate this model towards you, you would be looking now down through the glottis, which is that opening right there. Here's the trachea. So now you're looking from a superior view, kind of like the book. And so what I did was I zoomed in on that view of the glottis. Here's the glottis, that passageway, the little ribbed structures that you see are the folds of the trachea. And then what you can see now a whole lot more clearly are the vocal folds. So here would be those vestibular folds, and then just inferior to it would be here. Those would be those true vocal folds. I don't talk too much about vocalization. Your book does an okay job of explaining how that works. You can read about it in your textbook, but just kind of in general, what happens is air makes its way down, and it's hard because this is a two-dimensional surface, but air makes its way down into the voice box and down into the trachea, or up and out. And as that air passes by these vocal cords right here, they vibrate. And so when they vibrate, they are going to allow us to make a variety of sounds. Primarily what changes the vibration frequency of those folds is how thick or how thin they are. And so there are muscles that will stretch those folds all the way across here and thin them out. Or when those muscles relax, those vocal folds get thicker. The thinner those vocal folds are, the higher the pitch of the sound, and the thicker those vocal folds are, the lower the pitch of the sound. So these vocal folds are under the influence of testosterone. So the greater amount of testosterone that's circulating in the body will cause those vocal folds to become thicker. And of course, the opposite is true. The less amount of testosterone that's passing through the body will have a lesser effect, and therefore those vocal folds will be a little bit thinner. So we have to go back here because I put this. I've already looked at that slide. Okay, so now you're looking at a posterior view of, this would all be larynx up here. So really what we're focusing on is now a posterior view of the trachea, and that's right here. So the previous slide we were looking at an anterior view, and from a posterior view you can still see those tracheal cartilages, but what I want you to recognize is that they don't go all the way around. They're not continuous, that there's this open connective tissue portion of the trachea posteriorly. And there's a couple of reasons for that. First of all, the esophagus would be immediately behind or immediately posterior to the trachea. And when you swallow a bolus of food, that bolus is going to project into, sorry, let me erase that, that was dumb. That bolus is going to kind of push into the posterior part of the trachea, and so we want that trachea to be flexible. The second function that not having continuous cartilage rings around the trachea is that embedded deep to deep to this connective tissue that you see here is a muscle called the trachealis muscle. Let me get back. Oh my God, hold on. Let me get back to my pen. So there's the trachealis muscle. And I'll switch to red so you can kind of see. That trachealis muscle would go from the end of the C-shaped ring to the other end of the C-shaped ring, or the other side. It really essentially extends all through here. And its function is to change the diameter of the trachea. So if you imagine just as you're sitting now, the trachea has a fixed diameter, meaning it's only so wide. You're just sitting down, you're breathing normally, you're not exercising. But for instance, if you were up exercising and there was greater demand for oxygen to get to your body, that trachealis muscle would relax and it widens the trachea, allowing you to take more air in per unit time. So that's the function of the trachealis muscle. Another thing I want to mention before we move on to the next slide is that the trachea is lined with pseudostratified ciliated columnar epithelium. And remember, its function is twofold. Protection, because the cells, while they're not truly layered, they make a nice thick layer of protection over the underlying connective tissue. And secondly, here in the trachea, what that type of tissue does is it has goblet cells. The epithelium has goblet cells embedded between it, and goblet cells, as you recall, produce mucus. And the cilia on the surface of those cells move that mucus up and out of the body, and that mucus is going to trap pathogens, debris, microorganisms, whatever the case may be. So the goblet cells embedded between the epithelium make the mucus traps pathogens, and the cilia beat that mucus up and out of the body so it doesn't get down any further into the lower respiratory tract. Histologically, when we look at the trachea underneath the microscope, maybe one of the first things that you'll notice is here is the lumen, that's the passageway where air will pass through. And then you can see the thickness of the trachea here. So this would be the most superficial, right? And then this would be the deepest layer of the trachea. Now, we're not going to study the histology of the trachea in the same depth that we did the digestive system, but I do want you to recognize a couple of things. I want you to recognize the thickness of the trachea, and also I would like you to recognize the hyaline cartilage rings. I know we can't zoom in on this, but if you could, you'd see that all of this was hyaline cartilage. So that ring essentially, let me see if I can use a different tool here. That ring of cartilage goes from oh geez, hold on. That ring of cartilage goes all the way around. And so here would be the tip right here. So that's the end, and here's the other end. And spanning between those two ends, these are the strips of smooth muscle that make up the trachealis muscle. So you can see that going all the way over to the other end. So in this specimen, when the specimen died, you can see how it's kind of cinched up here in the back. Everything is kind of scrunched up here in the back. That's because that trachealis muscle was contracted at the time of death for this specimen. I know it sounds a little grim, but I wanted to point that out to you. This space here where you see that there isn't any hyaline cartilage is just because the knife happened to not cut through in a straight line, and it just missed that piece of the C-shaped ring right there. Okay, and also if you could zoom in, you'd see right here would be the pseudostratified ciliated columnar epithelial tissue that you would recognize from earlier study. You'll be able to see this when you look at it from the pictures either in class or on our MESA website or on histologyguide.com. So you would want to zoom in on there, right there, to see the pseudostratified. So from here now, we've just learned about the trachea. And now what's going to happen is the trachea splits right about here. And inside, there's a little ridge right there, and we call that the carina. So that's the official splitting of the trachea, and at that point, the trachea becomes structures called the primary bronchi. Bronchi is singular, bronchus is plural. So this would be the left primary bronchus. This would be the right primary bronchus. And one of the things you can see is that these primary bronchi still have cartilaginous rings. But you see how they're split right here and like right here? Those cartilaginous rings are going to gradually disappear. Not in the primary bronchi. You can see them beginning to disappear in the primary bronchi. But as we get closer to the gas exchange surface, those hyaline cartilage rings will eventually disappear. And I'll remind you of why that is a little bit later. The function of the primary bronchi are to deliver and remove air from each lung. So this would be the left lung, and the right lung would be over on this side. Now the right lung and the left lung are both divided into lobes, so I don't want you to be confused on that. Just know that there's right lung and left lung. We don't really say right lungs, plural, and left lungs, plural, because they're not all called lungs. I'll show you what I mean in a second. So some anatomical differences between those two bronchi. One would be that the left is a little bit longer. So there's the left primary bronchus, and here's the right primary bronchus. So you can see that it's longer, and it's not shown very well here, but the left primary bronchus is angled a little bit more laterally. If I had to draw it in, I would draw it like that. And the reason for that is because your ticker, your heart, sits right there, and so the left primary bronchus has to accommodate for the heart. The right primary bronchus is wider, even though it is a little bit shorter. Now, each one of these primary bronchi enter the soft tissue that we would recognize as lungs at a location on the lungs that we call the hilus. So let me get a different color out here. Maybe I'll use green. And in purple, all of this, that's the soft lung tissue, right? That's going to be made up of some connective tissue and structures called alveoli. But you see, I want to point out the purple lung to you, and then the point at which the primary bronchi enter, which would be like right here, we call that the hilus. I believe, if I'm not mistaken, singular is hilum and hilus is plural. And as far as the histology, the epithelium is still pseudostratified ciliated columnar epithelial tissue. Let's have a look at that. And one of the things that you can see here is, first of all, this section right here is totally devoid of hyaline cartilage. There's hyaline cartilage here, there's hyaline cartilage here, here, and off screen right here. You can still see parts of the trachealis muscle right here, and that's all cinched up in the back, but no cartilage, no hyaline cartilage right here. And so that's a sign that you're no longer looking at the trachea, that underneath the microscope you're now looking at the primary bronchus. Again, if you were to look at the epithelium, and you can kind of see it from here, that epithelium would be pseudostratified columnar, pseudostratified ciliated columnar epithelial tissue. As we continue down towards the gas exchange surface, those primary bronchi turn into or give rise to what we call the secondary bronchi. Secondary bronchi, so you know what we're looking at, are these structures here, 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 and that's really all you can see. Oh, right here as well. So the left lung, because you have the heart sitting right here, the left lung only has two secondary bronchi, because the left lung only has two lobes. So there are two lobes. Let me use a different color here. Let's just say there's a lobe of the lung right here, and there's a lobe of the lung right here. And so because there are only two lobes, there are only two secondary bronchi. On the right side, which you also can't see, you'll just have to trust me on this, there would be three lobes to the lungs. Let's just say one right here, there would be one in the middle, and then there would be one that's located a little bit more inferiorly there. So therefore, there are three secondary bronchi on the right. And another identifier here, as I mentioned earlier, will be that the cartilaginous rings, the tracheal cartilage, are now going to be very small pieces, plates of cartilage. So if I erase what I was drawing here, you can see how these hyaline cartilage plates are continuing to be reduced. We can look at a secondary bronchus underneath a microscope. You're not going to have to determine for lab you won't have to identify a primary versus a secondary bronchus in lab, but I'm just showing you how you can even tell underneath a microscope, where now you've got just these small plates of cartilage all around, rather than a full ring of hyaline cartilage. This epithelium right here would still be pseudostratified ciliated columnar epithelial tissue. Now we're moving on to the tertiary bronchi, and the secondary bronchi give rise to the tertiary. So let me show you what I mean. So here's move that out of the way. Here's the primary bronchus. That's this section of tube right here. And we just finished learning about the secondary bronchi, which would be here. And now these are going to be the tertiary bronchi. So here, here, here. And one of the things you'll notice again are these hyaline cartilage plates are just getting smaller and smaller, until when you get all the way down to here, these aren't bronchi, they're what we call bronchioles. But when you get all the way down to there, those hyaline cartilage plates are missing altogether. The tertiary bronchi make their way to each segment of each lobe. So remember, on the left side, there are two lobes, and those lobes are going to be broken up into segments. You don't need to know that for either lecture or lab. I'm just trying to make sure that everything is clear for you here. At this point, in those tertiary bronchi are still lined with pseudostratified ciliated columnar epithelial tissue, but now that's about to change. So as we move into as the air moves into the bronchioles is where we're going to see the first change from pseudostratified into now simple columnar, and then eventually simple cuboidal. Imagine, again, squeezing that epithelium, trying to get it as flat as possible. The flatter, the thinner the surface, the better for diffusion of gases. Here's a tertiary bronchus underneath the microscope, and all you really see left is just a little piece of hyaline cartilage here. Again, pseudostratified there. lining the lumen. So that's the end of the respiratory system part 1. And when we come back, part 2 will begin discussing the bronchioles with you. Okay? See you in part 2.